But what's most interesting from our brief conversation this morning is that not only is she doing killer work, but she actually has done 2 field seasons on a complete ACL tear that she needed to get replaced. So from Utah, a little skiing accident, but it didn't hold her back one bit in the field. So I'm proud to welcome the brilliant and the tough Sarah Bush.
They have some dark slides. Well, that's a great introduction. I've never been introduced as tough before, so that's awesome. So I have a mic for the camera. Instead of wearing 2 mics, I'm just going to speak up. So if you can't hear, please say so, and I'll speak up. Today, since I was told this is supposed to be a general audience, I'm going to give you a little bit of a flavor for some of the different projects I have, and then tomorrow I'll give you a little bit more of the meat of some of the experimental projects that I've been doing in the lab. So tonight what I wanted to do is take a fun approach to the work that I've done that enlightens how we think about ecology and evolution of parasites, especially. And you can't go wrong thinking about evolutionary ecology by starting with Darwin. It's great to think about this because we're 150 years past the publication of Origin of Species. And we have this fantastic framework, thanks to Darwin and Wallace. I did work in the beginning, so I have to include Wallace. In terms of putting this framework together and unifying biology in a central theme. We can use these patterns in space and time to bridge micro-evolutionary to macroevolutionary processes and ecological processes that influence evolution and influence macroecological patterns. So we can use evolution to understand how species are distributed through space and time. I'm really excited about that. One of the things, though, that I think gets a little bit overlooked is, although we have this brilliant conceptual framework, where this started for Darwin was in his own backyard as a naturalist, collecting beetles and other bugs, but also in his trips around the world, actually collecting organisms. And so everyone's familiar with his trips on the Beagle, and the specimens that he collected, some of them, such as this one right here, are still around today. They're in the London Museum of Natural History, and you can go and visit them. You can touch them. You can see the data that he recorded. They still exist. And like many of the other Victorian naturalists that were around at the time, there are other specimens, such as this one that was collected by Captain Cook's voyage, or ones collected by Lewis and Clark on their journeys across the country. These early geographical voyages that we think of as discovery were also biological in that they were collecting specimens that were being brought back to study. And they were really revolutionary for their time. So these are really exciting because they still exist and we can go back to them for many different reasons. So one of the things that Darwin did once he got back was he started revisiting his specimens, and these are the famous group of finches called Darwin's finches. And these finches, as well as mockingbirds that were collected from different islands, actually played a major role in how Darwin started to think about and conceptualize how natural selection might work. There was variation in these individuals among different islands, and thanks to his colleagues, like Gould, who actually took better notes than Darwin, he was able to look at where those birds were collected from different islands. Darwin noted that they were from the Galapagos, but not what island they were taken from, actually. So with the help of other naturalists, he was able to start to piece together some of these spatial variations and come to these great conceptual advances. And so from his notes before Origin of Species, we get this quote: "From an original paucity of birds in this archipelago, one species had been taken and modified for different ends," which is so incredibly insightful. Now, with modern techniques, we have the molecular tools to go ahead and reconstruct the phylogenetic histories of these organisms, and we can now more closely understand, or more accurately understand, how different species are related to each other, which ones are more closely related. We can track these patterns along with the patterns of biogeography in terms of which islands are older, and follow how these birds evolved over time. And I didn't put it on here, but there's now an even more recent one, that paper that just came out that shows the complete genotypes of all of these individuals and interbreeding, which is exciting, the amount of interbreeding between some of these individuals. Hybrid integration. So after all of these years since Linnaeus of discovery and naming specimens and this conceptual framework, what is the taxonomic status of organisms on the planet? Well, if we look at charismatic megafauna like these, we know that birds, we know a lot about birds. There are about 10,000 species of birds on the planet, and every once in a while we get a new species that's described, not just one that's a cryptic species that's already known, but something that's truly new. Mammals, we also know pretty well. We probably know 95% of the mammals on this planet. There's about 5,500 mammal species, not quite as diverse as birds. Reptiles, a little bit less well known, but still we know 87% of the reptile species that are on the planet. Of course, we don't know exactly because we're quantifying an unknown here. We don't know how many species, we don't know, but these are estimates based on species accumulation curves. Amphibians, we start to get at, they're smaller, they're harder to see, so they're collected less frequently. There are fewer known species than there are unknown species in this case, and I have colleagues at Kansas that are rapidly describing amphibians. They often do it by vocalizations, and the vocalizations of the amphibians are really important. And he works especially in the Philippines, which is where Armageddon Now was filmed. And he was watching the film and like, oh, that's a new species. And then he went and collected it and described it. So Hollywood actually recorded it first. So these things are happening with vertebrates on a fairly regular basis, but at a much lower scale for the things that we know a lot about, birds and mammals. In fact, there's an amphibian, a new species of amphibian that was published this week. If anybody's seen this, this is a new species of glass frog from Costa Rica. Just came out. My 6-year-old twins really like it because they think that Kermit finally found his relatives. Right, so these discoveries are happening. But what's my point? So I'm a parasitologist. For all of this amazing diversity that we're used to thinking about, especially when we think about biodiversity, like your favorite organism, there are many, many more of these. These are parasites in various forms of another. And they're much less well known. If we start thinking about the parasites, some of which that are on here, insects, we only know 20% of the diversity. We already described 1 million insect species. That means there's 4 million insects that we haven't described yet. Nematodes, there's probably 500,000 species of nematodes. We've described 5%. Platyhelminths, so this includes cestodes, trematodes, acanthocephals, a lot of the parasitic helminths. We've described 25% of probably 80,000 existing species. Now, not all of these groups, there are free-living individuals in all of these groups, but probably 30% to half of all of these groups are parasites. So we know very little about the existing parasite diversity. And so in many ways, we're still kind of on that voyage of the beagle for parasites. We still are in the area of trying to discover what's out there. So a large part of what I do in my lab is to collect, preserve, and describe these parasite species with collaborators depending on the group. I focus a lot on insect parasites, especially on birds. So I want to talk a little bit about that before I talk about some of the ecological patterns that I found. So one thing I should say, though, is I'm going to talk about collecting, and there's been some bad press in the news lately about vertebrate collecting especially. And one thing that you do if you're collecting parasites is you collect their hosts. And so I want to just put collecting in perspective for a minute. So a little backward, a little segue here. Why do we collect? Well, one thing that we do is when we're describing the species, we need a voucher specimen. So it's a typological system. We have a single individual that we select to represent the species. And we know that there are 100 different species concepts, really depending on who you talk to, and species vary in time, they vary in space, and it's a continuum. But we select an individual to represent what we're going to call that species so that we have something to go back to when other people start asking questions. Is this the same thing? Is it different? we can go back. We can go back to it morphologically, we can go back to it genetically, and that's one reason why a voucher specimen is so important. We also collect specimens in terms of series, so we like to collect multiple individuals so that we can capture variation that exists over space, variation that exists over time, and we can use that data to more rigorously understand the processes that are driving that variation. We collect tissues so that we can understand different aspects of anatomy, different aspects of hormones, different aspects of the physiology of these animals that we may not actually think about when we're collecting them, but other people who are interested in that can go back to the specimens in the museum, like a library, and check out the specimens and get what the information they need to know. We also collect behavioral data now using tools like recordings. I'm going to show you a few sound recordings and videos that we can associate with specimens in museums, or we can database on their own to get important information about how organisms interact. And of course, my favorite, parasites, which haven't been collected as much, but some of the old specimens, like Darwin specimens, we can go through and we can sample some of them, and in some cases still get parasites. The species of last one, the passenger pigeon, was collected after the passenger pigeon went extinct. So there's still specimens there where we can get parasites. What do we use this data for? Well, for those of you who are bird watchers or like field guides of any sort, field guides are a visual average done by artists where they go and they look at the variation in specimens and put that into a unified system so that you can study what you're seeing out there. So that's a great system that's available to everyone. Specimens are also available for scientific studies now and in the future. And a lot of times the data that's associated with the specimens we collect are used by management and conservation organizations to help prove that specimens or species are in an area, and they're used to designate national parks. In fact, some of the specimens that I collected in China were used in one of those things, and there's now a new national park in China that was done because of the collecting work that the Kansas group that I was a part of had contributed to. So if we have specimens of everything, why should we keep doing this? Well, the most famous example is probably that of DDT. And so the fact that people had collected eggshells in the past, and then once DDT was used, they were able to collect eggshells again and compare the thickness of the shells and prove, based on a continuing series in the museum, that that thinning was due to DDT, and it was able to ban the use of that chemical in the system. Without the museum specimens to prove it, it would have been really hard. We also have the future to think about. And there are people that are out there right now that are working on taking DNA from the extinct passenger pigeon and combining it with DNA from the extant bandtail pigeon, which is its closest relative, to try and bring this organism back from the wild. I tell them that they have to bring the parasites back too, but they're not listening. So I don't know, this is a little bit out there, but it could happen. And the future provides a lot more technical wonders that we don't even know are possible yet. So just to put, that's kind of the context of what we do in the field. And as a parasitologist, I add parasites to all of that. One thing to think about is that there have been, that we do sometimes catch birds and euthanize them and put them as specimens in the museum. Not all of the things that we catch are allowed to go. But I want to put this in perspective as well, so I'm going to ask you to think about how many birds do you think might be killed by striking windows? So that's kind of big. Let me narrow it down. So think about how many birds are killed by striking windows in one major city, say during fall migration. Any numbers? 100,000? Is that what you said? Okay, that's a little high for one city. Anybody else want to guess? I mean, I don't know the answer. But in Toronto, in one migration, 2,000 birds hit the windows and were recovered. That's just the ones that were recovered. I don't know what the answer is. There are studies in Chicago where they're trying to figure out what combination of lights on, lights off is going to help keep birds from hitting windows. And somebody else is looking at UV marking So birds can see in UV, and they're thinking about putting like crosses on all the windows so that the birds see the UV, but that we don't. So there's interesting technical advances that are going out for that. But to put it in perspective, in the US, somebody came up with an estimate of 35 million birds per year are killed by striking building windows, in addition to another 60 million road kills per year, in addition to hunting. These are massive numbers. This is the annual mortality of birds. US museums collect 15,000 specimens worldwide in a given year. So we're a drop in the bucket. So please don't worry about the dead specimens I'm going to show you. Okay, so in the 21st century, what do we do? Well, this is the HMS Beagle now. We get on this, and then there's the fun part of the job. I'm going to take you to New Guinea right now, and I'll show you some of the field work that we've done there. So this is an airplane that we've hired to go to this little landing strip right here in the southern part of New Guinea. Here's the landing strip. This is a landing strip where the pilot drops you off and says, get the goats out here to clean it out, or I'm not coming back in a month to pick you up. And so the goats go out and feed and clean up the airstrip. So when you get there, everybody in the village comes out. The fact that an airplane has landed is a major source of entertainment. We were greeted by the local kids who had their little airplanes to show us. And the plane, as we left, the plane was also taking a doctor out. This is the first doctor to ever visit this village. This is how remote it is. And he was doing the best that he could to treat the kids in the village and various ills. But the infant mortality rate in this village is 90%, primarily due to malaria. They have a certain height threshold that they don't even count people in the village unless they're pretty much full grown. These are the types of villages they live in. They're up high because it rains a lot. And this is the men's village with some boats underneath it. And I'll show you that that's a boat. And then we get our stuff off of the airplane and carry it to the boats. And in the boats, we load everything in and we carry it across the river up into this forest. This is where our field site is. And these boats are about 100 years old and they have holes. And they say, oh, just here's some mud, patch the holes fast. Right? And you patch them really fast and hope you get to the other side. They don't seem worried about it, but having holes in the bottom of the boat seemed to concern me. But we got across both ways. This is the bottom of the forest, and this is where we're headed. We're headed up here into the cloud forest. This is an area in New Guinea where very few people have been. It's an area with really pristine forests, and very few of the birds have been studied here. When we're in the field, we set up tarps to keep us dry. This is our base camp where we lived for 6 weeks. And we immediately start setting up traps. So this is a mist net. How many people in here have used mist nets before? Anybody? Okay, so you know they're like badminton nets. But in the rainforest, there are species of birds that only fly in the canopy. So a mist net on the ground is not going to catch them. And we were really interested in getting some of these canopy species. So right here is one of the mist net poles. We tied the poles on a pulley system. And here's the other pole right here. And the mist net is going between there. Unfortunately, the guys in New Guinea are good at climbing trees with machetes and are able to get up there and help us set these mist nets. So we had mist nets in the canopy as well as some on the ground. And birds fly into these mist nets, which is this flycatcher here, and you can pull them out and remove them if it's a species that you've already sampled thoroughly, or you can keep them for further processing. So here's some birds that were caught in the mist nets, and these are the locals that were good at helping us. And in New Guinea, unlike any place else I've ever worked, these guys know the forest. They live there. They're really interested in all of the birds. They'll tell you which ones taste bad. They'll tell you which ones taste good. They'll tell you which ones not to lick because it makes your tongue numb. There's poisonous species of birds there that are really fascinating, and they're really good at hunting them. So these are some of the specimens that we got. But in mist nets, it's good for catching small birds, but there's also some megafauna there that we were interested in sampling. And by megafauna, I mean big birds, so cassowaries. They're like the size of emus, right? But they're meaner than emus. In fact, cassowaries are birds that have killed more zookeepers than anybody else in the world. And they do it by disemboweling them. For example, this is a cassowary. Now, in New Guinea, they're pretty skittish. If you see, I've seen some in the wild, and it's normally just the tail end of them like running away very fast. But the guys in New Guinea use cassowaries for bushmeat. They especially like to hunt them for big ceremonies like weddings or rites of passage. So they do catch them occasionally. And we were fortunate, I'm going to back up a little bit. So because they're megafauna, they're also susceptible to being endangered or threatened. They aren't yet, because the forests here are in pretty good shape, but we're keeping an eye on it. And they're hard to catch because they're so big, and you wouldn't really want to hold it down while you looked at it for lice. But you can do a lot of things without doing that. And one of my colleagues, Andrew Mack, spent his thesis work and many decades in New Guinea studying cassowaries, and he did it by studying cassowary dung. So this is a pile of cassowary poop in the forest, and the New Guinea word for that is pek-pek. And you can get DNA from that, and you can also study their diet. And so he was really interested in how cassowaries play a major role for seed dispersers. And when we lose cassowaries, you're going to lose the big seed dispersal agents in New Guinea. There are big seeds there that are very large, and cassowaries are the only thing that can disperse them. And so he was really interested in studying that. And he was also interested in tracking populations by looking at vocalizations. So here's some of the data that he has on vocalizations. And the interesting thing is that the recording equipment that they ended up using is stuff that was designed for whales because it's really low. And in the rainforest, you might as well be in the ocean. It's really wet. So let me just play this for you. So you get to hear that in this kind of forest. One of Lopez actually did catch one, which he chased through the forest and speared. And they were barbecuing it, but before they barbecued it, I was able to get some specimens, including this new species of tick, which was found all over the head of this particular cassowary. And we also sampled the skin because Richard Prum, who's at Yale University, is really interested in studying the physics behind color. And blue is a really hard color. It's not something that you can get from your diet. It's a structural color. And so it's a really good indicator of host condition. And this type of tissue is not something that's very common in the bird world. So he was very happy to get those specimens. One of Rick's students is Ed Scholes, who I was in New Guinea with. And he studies sexual selection, including blue coloration, but on a more behavioral style scale. He is interested in birds of paradise. And we'd go in the field, and he'd set up his blind to film these birds. And they've got a whole National Geographic thing on him now, which is fun to watch. But he let me use one of his videos, if I can get this to work. So this is a bird of paradise. There we go. This is a male, and it's cleaned an area. It's waiting for a female. Here comes the female. And they do this amazing dance, and he's looked at the evolution of the dance and the different patterns that they exhibit. They have these really iridescent feathers. I mean, how did this evolve? This is just nuts. It's like an alien, right? And we had lots of arguments in the field. And I told them that this is all because of parasites, right? This is sexual selection in its peak. This male is trying to show the female how good its genes are. Feather lice, they reduce survival, they reduce energetics. It costs a lot of energy to get rid of your parasites like lice, and so males tend to display less. They also have poorer feather quality. They chew the feathers, and so those iridescent feathers aren't going to look so pretty to a female. So that gives me my segue to lice. So when we're in the field, we process birds for parasites. So here's a New Guinea pheasant pigeon, and we've been sampling the lice off of this particular individual and something else over here, I can't tell just what. So we sample the parasites, and then we look at them back in the lab. And these are some feather lice, some lice from birds, and I just want to point out how incredibly variable they are. They're not the same thing. They may just look like specks to start with, but once you start looking at them, they're incredibly diverse. Some of them have really triangular heads, some of them have long skinny bodies, some of them are short and fat. Notice how white this one is and how dark this one is. Tomorrow, I'm going to talk about the evolution of cryptic coloration in these guys. This white louse is from the white sulfur-crested cockatoo. This dark louse, photographed here on its own host feather, is from the yellow-tailed black cockatoo. And we see a pattern of repeated evolution of cryptic coloration in feather lice, just like you might expect if you've studied your Bactellaria in the field. And so I'm not going to spoil the details of that, but we have a lot of experimental work to follow up from this field work on the evolution of cryptic coloration that I'll talk about tomorrow. So one of the other things that we get to do, though, is that we get to sample lots of different birds. When you're in the field, you're struck primarily by the fact that most of the avian diversity are songbirds. There's 5,000 species of songbirds, and most of them have lice. We haven't sampled them yet. We only know about 300 species that have been described. But part of this is because there are thousands and thousands and thousands of species in museums that nobody's bothered to describe because the 300 species are a total mess. We don't know how they're related to each other. They vary in size, they vary in color, they vary in shape, and we know that they're all kind of together, but depending on who you talk to, the revisionary systematics of this group is insane. So with a colleague of mine, Kevin Johnson, who's at Illinois Natural History Survey, we decided to tackle it with a molecular approach and a morphological approach at the same time. So with all of this collecting, we had fresh samples where we could sequence material. So we collected fresh sequenceable material from 275 different host species. And we sequenced them, mitochondrial DNA and nuclear DNA, and we built a phylogenetic reconstruction. I'm going to show you the tree, but I don't want you to be scared. I don't want you to actually tell anything about this, but this is the tree that we built. Did you catch all that? It's diverse. And the interesting thing is, of all the specimens that we sequenced, 10% of them are described species. The rest are new. So it was a much bigger project than we had anticipated. And so I hired a postdoc to help with all the descriptions. And the first thing to know when you hire a postdoc to do taxonomy is get one that wears a pink helmet, because he's great. So we have 39 species that are known in the phylogenetic reconstruction that we built. And he's working on identifying and describing the other 250 unknowns in this particular group. But there's some really interesting patterns that have emerged that I can talk about at this point. One thing that is really interesting is this is enormously diverse. Why is this so diverse? Well, one thing that happens with parasites, and especially lice, is host specificity seems to be a major driver of diversity in parasites. And just to give you a little bit more information about lice, lice are obligate ectoparasites, so they're always on the host. They can't live off the host. They can't fly, they can't jump. They're permanent ectoparasites. And so the only chance lice get to move between host individuals is really between mates or between parents and offspring in the nest. And so when you think about lice and their divergence, the classic tendency is to think about the hosts diverging and speciating and the parasites following suit. They diverge and they speciate with their hosts. And we definitely find that pattern in lice. It's especially true in gopher lice. There's classic studies on that. And we find it to varying degrees in avian lice as well. And still, the jury's out on this because we're still trying to resolve parts of the Borrelia phylogeny to see how that relates to songbirds. We're still in the early stages on that. I expect that we're going to have bursts of co-speciation that are driving this. One thing we can do is we can look at host specificity And amongst all of the Borrelia that we looked at, these lice are found on 1 to 2 host species on average. You don't see a single louse found on lots of different hosts. It's usually only on 1 or 2 host species. And those host species are usually closely related. So host specificity does seem to be important. However, there are some interesting exceptions. And the most extreme case is this louse, which is Borrelia docophoretes. And all of the lice in this clade are the same species. At the most conservative level, those are the same species. Depending on your molecular cutoff, some of the more liberal analyses would include all of these samples as a single species. This louse is found on a minimum of at least 18 different host species in 11 families and 20, maybe as many as 27. So this one is not host specific. 85% of the lice in this group are found on a single host species. This one is the extreme opposite of that. How is this possible? I mean, things that would never ever come in contact. Well, it turns out the Borrelia also have this really interesting thing that is a behavioral thing that they can do. So this is a hippobossid fly, an SEM of a hippobossid fly. And these are Borrelia lice that have hitchhiked a ride. They're basically using this fly as a taxi cab to get to the next host. Flies, since they have wings, are much more mobile than mice, and they frequently move between different host individuals because they can get there on their own power. We found flies with as many as 30 individual lice attached to them. I don't know if that fly can get off the ground, but the lice certainly attached to them. Now, if you look at all of the records of pheresis, and there are some, especially in older literature, where people just noted this cool natural history fact, virtually all of them are lice in the genus Borrelia. Not all of them, but virtually all. So clearly, this is a unique behavior that some of the lice in this group have really latched onto, and maybe it's that key innovation that helps them become a generalist instead of a specialist in this system. So this is something that warrants a lot more work, and we're just starting to get into that. Another thing that we've noticed, so this is a cartoon of a giant phylogeny that I showed you before, but scrunched into one side. We see a lot of different morphological variations within this single genus. We have kind of short round lice, some that are really long and skinny, some that are really short and fat with broad heads, some that are bigger with more triangular heads and lots of setae. This is all within a single genus, if you look at it molecularly. It's really odd. Most lice, the genera, are fairly uniform in terms of their morphology. Well, we know a little bit about the behavior of these different forms. These are what I would call ecomorphs. This is a head louse. It has a really big triangular head. These wide temples support really large muscles, and the louse uses it to hang on to the head feathers of the host where the host is going to scratch, and that scratching would dislodge that louse. And presumably those large mandibles would be able to help it hang on and avoid that kind of host defense. These other three types of lice are found on the host body where they are susceptible to preening. That's when a bird uses its bill to remove debris and ectoparasites from the plumage. This louse is a wing louse, and it inserts its entire body in between barb spaces on the wing. It's kind of like someone lying between two railroad ties to avoid an oncoming train. This louse can slide in between those barbs and avoid preening that way. This louse is a body louse, and when birds start to preen, they burrow into the downy feathers on the body of the host. And this is more of a jack of all trades. We're not exactly sure what it does, but it seems to kind of be in the middle of everything. And certainly it is known to run around the body of the host. So if we use these morphological concepts or ecomorphs and code them on the tree, or let me back up. This ecomorph concept and apply it to the evolution of this group, it's a lot like lizards. So those of you who are familiar with Anolis lizards in the Caribbean, they are broken into several different ecomorphs. And what happened is the lizards invaded different islands in the Caribbean, and they found a niche. Additional colonizations of the island later, new lizards arrived, they competed with each other, and they experienced character displacement, where one would specialize on one microhabitat and the other. And then on some of these bigger islands, after repeated colonization events, what happens is you get this somewhat closely related radiation of lizards where you have things like crown giants that are large lizards that specialize in the tops of the trees, all the way to little things that specialize in catching insects in the grass and the bushes down below. Well, the same thing seems to be happening with feather lice. We have feather lice that are specializing on the wings, the heads, the body, and kind of more of a generalist that's able to move around. And it's probably repeated colonization events, such as those with phoretic dispersal on flies, that are leading to this possibility of coming onto a new host, competing, character displacement, driving this variation. And I've done some experiments with competition amongst these species, and we do know that there is competition, and it does lead to character displacement. I heard that Bob Holt was supposed to be here, which would have been awesome because I would love to show him this data. It'd be fun to talk to him. I think he would really like it. But I'm not going to talk about the competition stuff right now. So I think it's a similar process of adaptive radiation, similar to what we see in other systems. If we look within the genus Borrelia, what we see is repeated evolution of these different forms. So we have generalist forms that crop up throughout the tree. We have head lice that crop up in various places, wing lice that crop up some places, these red ones. So morphologically, they're fairly labile. We're seeing convergent evolution of these forms. This is based on morphology. I'd like to get a lot more behavioral data. We've got to do that and figure out more about the behavior of these lice. That's the next piece on this one. So far I've talked to you about different processes that I think are driving the evolution of diversity in the system. Next, I want to touch a little bit on some factors that I think will be contributing to the loss of this diversity. So there are a lot of species that are listed as threatened. They tend to be charismatic, keystone species. There is one species of parasite that's listed by IUCN as threatened. It happens to be a louse. It's a louse on a pygmy hog in Africa. But I think that there's probably more than one parasite on Earth that's threatened. Just a wild guess. There are studies that have tried to estimate this. Co-extinction, these parasites, and especially lice, but there are other species that are just as specific, that should go extinct when their host goes extinct. Indeed, Jared Diamond considered co-extinction as one of 4 major drivers of extinction on Earth. There are some other studies that have looked at the relative host specificity of parasites and other mutualists and symbionts to try and quantify what the affiliate loss will be, because not all affiliates are parasitic, right, when species go extinct. And they looked at the affiliates on species that are already considered threatened and endangered, and they're estimating that at least 6,300 species of parasites should be considered endangered. And a more recent study by Dunn and his students focused specifically on endangered carnivores in North America and considered the specificity of those parasites and estimated that parasite extinctions are probably going to be 10 times that of their host. So I think that these predictions are all very useful and telling. But one thing that I also think is going on is that the parasites may be preceding their hosts into extinction. If we have 10 times as many parasites on a host on a dwindling population, don't you think they would be going extinct first? And the reason why I think that is this. Parasites tend to have a fairly aggregated distribution. They're not equally spread amongst the population. And so if you have a stochastic process that's reducing the host population, in some cases, by the time the host population becomes reduced to a small size, even if they're still around, you're going to lose the parasites. And nobody's really looked at this. So I used some field work in China as an opportunity to do this. It's a comparative study looking at parasite species richness in different size forest fragments. So here's the particular study region. This is one of the biodiversity hotspots classified by Conservation International It's right along the Vietnam border, so it's in this tropical area of southern China, which is a great place to work because very few people have been able to work in there. But the political doors are such now that it is possible to get in there. So I sampled parasites on birds at 4 different sites. They are all listed here, but importantly, in each of these cases, this is a circle with the base camp in the center and a diameter of 10 kilometers around the base camp. So this is where the nets would have been placed. And the dark areas represent the forest that is in that area, based on Landsat images. And you can see that in some areas there's not much forest, and in some areas there's a lot of forest left. Put this in yet a different format. The forest size range from Shipu, which is only 3 square kilometers in this area, all the way up to Shuandishan, which is 215 square kilometers in the same amount of area. And these boxes are to scale, so that you can visualize the difference in forest size. I didn't try to estimate exact fragmentation patterns in these areas because I don't know how the birds respond to them exactly, but the fragmentation also correlates with this. So in this area we went out, it was part of a general biotic survey and inventory project. So we surveyed a lot of animals in this area. And we surveyed 155 species overall and almost 1,000 birds in these cases. And for these individuals, we did parasite workups and sampled especially their ectoparasites, lice, ticks, mites, fleas. Mites are pretty hard to quantify, so the quantitative data is based on the larger macroparasites, fleas, lice, and ticks. What I did was, instead of trying to take data from each of these individuals, I wanted something that was very statistically robust to looking at parasite data across these different sites. So I focused only on the 7 most common genera in these sites. And I needed specimens where I could find at least 12 individuals based on species accumulation curves, parasite species accumulation curves. I need at least 12 individuals to have a 90% chance of getting all of the parasites in an area. I also wanted specimens where I could compare them from a small site to a large site. And once I used those criteria, this is what was left after all of the specimens that we sampled. 7 genera where we had robust enough information for comparisons. So this is a little different sampling strategy than most people are used to. So I tried to show it graphically here. So for each of these 7 bird genera, the birds were collected from a small site and from a relatively large site. For each genus, they were collected in different sites because plants and different things vary at each location. And so for Alcipe, it was comparing Shipu and Quangkoshi, whereas for Phylloscopus, it was comparing Shipu to Shuandishan. Again, it's all relative, and we're looking at the relativity of size in the analysis. But every bird, the number of ectoparasites, such as lice, from small and a large site. And here are the data on this. So in each case, the large forest had a higher parasite species richness than the small forest, and for 6 of the 7 host genera. For the 7th one, there was a reversal where there's actually a few more species found at the smallest site. We used a generalized linear model to analyze this, taking into account forest size and different aspects of the hosts themselves. But forest size came out as a significant predictor of parasite species richness. And this is really amazing. These are common birds. These are really common birds. The only way I could do that is to focus on birds that are easy to catch. The species that are hard to catch are already extinct. Their parasites are already gone. These are the ones that nobody's worried about because they're so common. But what this is indicating is that their parasites are already suffering. Another interesting pattern that emerged is that this is the same data but shown in a table format to make one specific point here. And that is in cases where the small site had a parasite and it was absent in a large site. So the reverse of pattern where you expect fewer parasites on a smaller site. The 2 cases where this happened tended to be ticks. So this species of tick is also found on chickens. And so it's probably the fact that some of the birds have parasites in a smaller site may be due to the fact that they're picking up parasites from domesticated animals like chickens, which is particularly problematic because those general species tend to be vectors for more diseases. Ticks carry a lot of rickettsial-like organisms and other bacteria that can cause sickness in animals, especially that aren't used to it. So not only are we looking at a difference in parasite species richness just in terms of value, there's a quantitative difference in that the parasites that are there are generalists and not specialists. So just to summarize this, we found that bird louse species richness is smaller in the forest fragments, smaller forest fragments, and that the implications of this are that if you want to conserve all of the organisms, and I know parasites aren't cute, but they're still organisms. If you want to conserve diversity for the sake of diversity and all of the ecosystem processes that parasites include, or other symbionts or mutualists that we're not studying when we study the vertebrates as a whole, we may need to protect areas that are much larger than what are needed to protect the vertebrates themselves. It also suggests that parasites maybe we might be able to use them as indicators of host health. I put a question mark here because I think a lot more needs to be done, but this suggests that you might be able to go into an area and instead of trying to do time transects in an area which is really time intensive, takes a lot of manpower to survey the host habitat, the host status, you might be able to go in, catch a few, sample their parasites. Ectoparasites you can sample without injuring an animal, you can let them go, and determine if the species richness of the parasites in that community are going up or down, and use that as an indicator of host population as a whole. So, when you're thinking about conservation setups, you need more legs, right? So with that, I'd say thank you for coming. Be happy to take any questions.
Well, while you're thinking of those questions, I'll announce that Ted, who has one, is hosting our social, and it's written on the board, and that'll be shortly after we talk. So please put that in your phone, get directions, and the usual Ecology Center seminar social will follow. Sorry to interrupt, Ted.
Well, I was wondering if you see quite a lot of variation in the number of individuals of these different species of parasites on individual birds. And is that another piece of data that you play with in looking at forest fragmentation and things like that, or competition and so forth? How many of these parasites will you find on a bird?
It depends on the size of the bird. Small
Yeah.
birds have few. It's incredibly aggregated, so you'll have an individual that has lots and many that have none. I actually looked at the relative intensity, so the number of individuals of parasites per host, to see if there was a difference. Because you might think that in an area where birds are stressed, maybe because the habitat is smaller, that the time that they spend to get rid of these ectoparasites, like preening, takes a considerable amount of time. They might not have the time to do that, and that their parasite loads would go up. And I didn't find any significant differences in intensity in this system, just species richness.
But when you say lots, do you mean hundreds or dozens or?
On these small birds,
Yeah.
probably in the 5 to 10 range for lice. But if you get to something that's the size of a feral pigeon outside, it would go from 0 to 11,000. So the range is enormous.
So on the forest birds, what's the fecundity of the lice?
Most of these lice lay a single egg every day, and their generation time is about 25 days, 30 days. So they can get loaded pretty quickly.
Yeah.
Birds spend it varies depending on species, but they spend probably 10 to 20% of their time preening, and that's absolutely keeps these lice in check. On a healthy bird, you don't see very many. If you see a bird that has a bill injury, if it's chipped or it's broken or it's deformed, you get lots of lice, a lot. And you'll have something you'll have a small bird with 100 lice on it. But it's the weird ones. Yeah.
I forgot what I was going to ask you. I got so excited about the next questions. I was going to, one question that's kind of a silly question is, do these lice have parasites?
Yes, they have parasites. They have a fungus that grows in them, and it's a hyperparasite, and it will grow underneath the sclerites and send out hyphae. We had it we have a culture of birds that are infested with lice in the lab, and we had it get in the lab once, and we managed to clear it by drying everything out. And that's the only ones I've seen. I know that there's a guy in Sweden who's trying to get these hyperparasites, and they're hard to find. But yeah, they do have.
So while we're talking about the actual parasite, it seems like there's a bit of a taxonomic barrier to entry in this line of work. I mean, do you care to comment on the importance of that type of training to get into these kind of evolutionary ecology questions?
I think that regardless of what you study, understanding the natural history is absolutely critical. And I think that not enough people understand the natural history of their organisms. You have to know what the species is. My postdoc is not here, so I can say this. I don't think you have to know exactly what it is. I think you can use shortcuts depending on your question. Barcodes, phylogenetic analyses to get at that. And certainly I use molecular techniques as a shortcut to try and, you can't look at 300 different specimens and in your head figure out how those are related. Especially when you've got convergent evolution of multiple types. You can't do it. You can take pieces of those and find smaller groups that you can work on and then do it and probably be fairly accurate. When we compared the molecular operational taxonomic units at some normal cutoff for insects, it actually lines up really nicely with the historical taxonomy in terms of actual species units. In terms of the greater taxonomy, that part was messy because people didn't understand how the groups fit together. So I think depending on the question, it is possible to take a few shortcuts. But I think a lot of the really interesting things that happen are when you understand how those molecular pieces fit together with species, how that, if they're morphologically different, how does that relate to how it ends up on the host? How does this last host interaction drive that difference? And we're also doing some biological species experiments in our lab now where we're taking 2 different species and putting them together and see if we can get a breakdown in reproductive barriers, as well as using different bird morphologies to drive the evolution of morphology in lice and trying to create species, basically, by causing differences. So I think that those are really interesting questions, and by not understanding the taxonomy, I think you miss those. That was kind of a long answer, sorry. Yeah.
Well, I'll dovetail on that. So you've been associated with museums of natural history. Do you care to comment on the importance of those organizations in furthering basic science? And I mean, we know that budgets don't necessarily rise for these organizations over time; they often fall. So any comments?
They're absolutely critical. It's like getting rid of a library. There's just so much there that we just don't know that we're going to need, and it's just a gold mine. We can't put enough value on it. Yeah.
I was interested in your story about the birds of paradise. I wonder, so has it been observed that in birds that do have these fancy displays that there might be selection for sort of morphological or, sort of phenotypic traits that are more louse unfriendly, so that you end up in a situation where you have sort of this interesting, the cascade of louse evolution, bird evolution.
I think that's a really, really interesting idea, and we don't have any evidence, but I think that maybe the fact that you have sexual dimorphism in and of itself could be really interesting. Because if you have species of lice that are evolving cryptic coloration, they can't be cryptic on everything else. It's not possible. So if you're getting a lot of transmission between mates, then maybe that one sex is more likely to exhibit its parasites. And we do know that with things like grouse that have the big air sacs, if there are more indications like hematomas that there have been ectoparasites that feed on and cause those, that they're less attractive. So we know that it has sexual selection piece, but I haven't tried it in the mice. Yeah.
So, to what extent do the parasite communities differ on these toxic birds? Do they have parasites that are resistant to those toxins? Do you know
Yeah.
anything about that?
Yeah, so they do. It can take lives from the pitibuis, so pitibuis are this bird in New Guinea that have the same, they've got homobotrichotoxin, which is the same toxin that the poison dart frogs, and they get it from the beetle in their diet, and it sequesters it into their feathers. And there are feather lice that live on pitibuis, and if you put lice from some other species on pitibui feathers, they die pretty quick. If you put where the pitibuis lice are more able to withstand them. Clearly they're used to them because they live on these birds all the time. The tests they're done in petri dishes, and I know Jack Numbacher was trying to do stuff with captive animals. The thing is when you bring them into captivity, they lose their toxin. And so it hasn't progressed much more than that. But yeah, no, I think there is this evolution, kind of coevolutionary arms race with the toxin in that system. And we do know that lice are able to evolve resistance to toxins because anybody in here who's had head lice and has tried to get rid of them with shampoo, it's really tough now. Human head lice have not, in fact, just one resistant gene. They've got triply resistant mutations now that confer resistance against pyrethroids. So we know it's possible, but it hasn't been worked out very well in the system.
I urge you to bring any other questions to the social, and let's thank Sarah one more time.